Obviously, the nervous system is a pretty complex structure to understand. So one thing that makes it easier is to learn about how the brain develops during embryogenesis. So basically, without getting into too much detail, the overall development of a vertebrate embryo looks like this. So pretty much all vertebrates, all animals, start off as a just a ball of cells, um, and then eventually called the blastula, and then eventually that ball sort of indents inward a little bit, and you end up with a structure like this called the gastrula, and that little pocket there will eventually become basically the lining of the digestive system. And in fact, at this point, there are actually two layers of cells here. So we have the outer layer, which is with the original lining of that blastula, and then this inner layer, and then eventually the whole thing grows into a tube. So you end up with something that looks like this, where there's an outer layer, an inner layer that forms a tube that goes all the way through. That's eventually just going to be the gut tube. So you're kind of seeing a cross section here. And then there's a layer of cells in between. And each that grows after the other two. And so each of those layers has a name. The outer layer is the ectoderm, and that just means outer layer. So ecto means external, and then derm means layer. That inner layer, that will be the lining of the gut, we call the endoderm. Endoderm. And then we have the layer in between the other two that we call the mesoderm. And so the entire body develops from those three layers of cells. The innermost layer, the endoderm, mostly it's going to form the lining of the digestive system and things that are attached to the digestive system, including things like the liver and other internal organs. The mesoderm eventually develops into most of the structural parts of the body, so things like bone and muscle, but also things like the heart and kidneys as well, and parts of the reproductive system. Meanwhile, the ectoderm forms mainly two structures. One is just the outer layer of the body itself. So for us, that means the skin and all of the connective tissue associated with the skin. And then the other major structure that forms from the ectoderm is this thing called the neural plate. And then the neural plate develops into two main structures, a neural tube, which will turn into both the brain and spinal cord, so the CNS, and then specialized cells called the neural crest that form eventually the cells of the peripheral nervous system. So here's kind of what that would look like if you could follow the development of an embryo. A lot of times when you see drawings like this, which you're probably seeing is a chicken embryo. So chicken embryos are really easy to follow because you can follow their development in the egg. And so this is kind of what a typical, pretty much all vertebrate embryos look like this. But again, this is still the blastula stage, so it's really just a ball of cells. It's kind of starting to elongate a little bit, but what you can start to see at an early enough stage, and the actual stage depends on the species, but here we've got kind of a thickening and, but more importantly, a groove starting to form down the middle. And so this whole area kind of marked in green here is going to be the neural plate. And you can see now here they've taken a cross section of that, and you can see that the ectoderm, that blue layer, is just one layer of cells. There's a layer underneath called the mesoderm, and a layer underneath that called the endoderm. But the rest of the ectoderm, all the stuff in blue here, will eventually turn into the skin, and then this region we're calling the neural plate will turn into the nervous system. So that neural plate, again, it kind of thickens and a groove starts to form down the middle called the neural groove. And that neural groove gets deeper and deeper, and the sides of that groove get taller and taller, and so we call those the neural folds. Meanwhile, underneath, other organs are forming. The mesoderm is starting to thicken along that line. That's actually what kind of pushes up the neural folds. And the whole embryo starts to elongate. So now, instead of just being a ball, now it's more of a long tube with one end, which will eventually be the head, the rostral end, and another end, which will eventually be the tail, which we'll call the caudal end. But the point is that again, that neural groove gets deeper and deeper until the two sides, the neural folds, meet in the middle. And instead of being a fold or a groove, it becomes a neural tube. So now you have just a tube running all the way down the length of the body from the head to the tail. In this case, you can see it kind of mid-formation. So the tube has started to form in the middle and it hasn't quite sealed at the head or tail end. In fact, there are birth defects associated with conditions where the neural tube doesn't quite close. So they're called neural tube disorders. An example of that is called spina bifida, where essentially the caudal end of this tube, which will be the spinal cord, doesn't quite close up, and it allows things like CSF to leak out, and it causes all kinds of developmental problems. But what's supposed to happen is this tube eventually completely closes up with a single tube going all the way down. These neural fold cells form a structure along the top where they meet. The mesoderm again gets thicker and thicker, and it starts to form these little bodies called somites, and those become more and more visible. Eventually, the somites show up as these little bumps along the sides of the neural tube. Those are going to be important for, again, the skeleton and muscle. In fact, the somites are the structures that eventually develop into the vertebrae, the bones that surround the spinal cord. But as that neural tube gets more developed, the neural fold cells, the cells that were part of that neural fold, eventually get covered by more ectoderm cells, which again, that'll just turn into skin. And then those neural crest cells now are where that neural fold was. Like I said, those will turn into the cells of the peripheral nervous system. Meanwhile, the neural tube will form into the cells and structures of the central nervous system. So that just means the brain and spinal cord. And then, of course, in the meantime, underneath you have all the other organ systems forming as well. So the neural tube, again, is exactly what it sounds like. It's just a long tube that stretches from the head to the tail. And as the embryo grows, the tube starts to grow and sort of enlarge at one end, and that end will be eventually the rostral end, which means it will eventually turn into the brain. And so the swellings that start to form at the rostral end of the brain, there are usually three of them. The most rostral one, the one all the way at the front, we call the prosencephalon. And so encephalon just is another long name for, actually it really means head, but we also call it the forebrain. So it's that region, that rostralmost third of this swollen part of the neural tube, we call the forebrain. And then just caudal to that, you have another swelling that here we call the mesencephalon, but the shorter name for that is just midbrain. And of course, that's what mesencephalon means. It just means the middle part of the brain. And then finally, you have an enlargement called, long name is the rhombencephalon, but hindbrain is an easier term to remember. And then everything past that, everything from here down, will eventually turn into the spinal cord. But we don't call it the spinal cord yet. It's still just the caudal neural tube. The forebrain is the largest and most complicated part of this neural tube. So it actually first divides into two parts. At first, what happens is the sides of the forebrain start to swell outward and sort of back like so. And as they get bigger and bigger, we call this region, these two enlargements, the telencephalon. telencephalon just means kind of end brain. And eventually, these will turn into the cerebral hemisphere. They get bigger and bigger and start growing kind of backwards and folding up over the rest of the brain. But again, they eventually form the cerebral cortex. And so in the adult brain, of course, this is what the telencephalon turns into. In humans, especially because the cerebral cortex is so large, it pretty much ends up covering the entire brain. And so the only way to see the rest of it is to look at the ventral surface, so you can see the other parts of the neural tube underneath there, or to look at it in cross section. So all of this this area is the telencephalon, or the cerebral cortex that forms from the telencephalon. And then in addition to the cerebral cortex, there are some parts that are underneath the cerebral cortex that are still part of the, are not part of the cerebral cortex, but are part of the telencephalon. Collectively, these parts are just known as the basal forebrain. So again, in the adult brain, or in the embryonic brain, you can't really see it. But in the adult brain, these regions become more prominent. So this whole area here in pink, not that region in purple, but this area in pink is all part of the basal forebrain. And there's some important things in there that we will talk about later. The other thing that forms inside the telencephalon are the lateral ventricles. In fact, as the neural tube expands outward, the empty space that's inside the telencephalon, or what we call the telencephalic vesicles, just grow into the lateral ventricles. Meanwhile, the part in between the two telencephalic vesicles, or in between the cerebral hemispheres, we call the diencephalon. And in fact, the word diencephalon just means the between brain, because that's where it is. It sits between the two much larger telencephalic vesicles, and they're eventually covered up. So as the telencephalon grows, the diencephalon becomes pretty much covered up by the telencephalon. It's a little bit easier to see in cross-section. So here's the cross-section of the forebrain. You can see, again, the two halves of the telencephalon growing outward. And then the diencephalon is just the part that kind of sits in between the other two, between the two telencephalic vesicles. And what's important, or one important difference between the telencephalon and the diencephalon, the telencephalon, again, has two halves, each of which has its own telencephalic vesicle, which become the lateral ventricles. Meanwhile, the two lateral ventricles, or what eventually become the lateral ventricles, meet and are joined in the center of the brain. And this cavity here, where they connect, eventually we call the third ventricle. But the area surrounding the third ventricle, these structures here and here, form important parts of the brain. So this region on top, there's two halves to it, is eventually going to become the thalamus. And then the region below, down here, actually both of these together, will form the hypothalamus. And in the adult brain, again in cross-section, here's one view of those two structures. Now here it looks like they're very small. This is just sort of the very anterior thalamus. And here's more anterior view of the hypothalamus. If you looked at a cross-section that was kind of further back, they would look bigger. But this is a nice area to look at, because you can see in the adult brain how the lateral ventricles both meet here in the middle, and then meet where they converge. This area is the third ventricle. And so basically the third ventricle is lined by the two halves of the thalamus and two halves of the hypothalamus. And so we'll come back and talk about both of those structures more later too. And then some other important structures that come out of the diencephalon are some of the cranial nerves. So actually all of the cranial nerves come out of the neural tube at some point, but the forebrain has at least two important ones. You have the optic nerves, which actually just kind of grow out of the walls of the diencephalon, like so, and they start as basically little cups that stick out. So you can see here, this is just looking at the wall of the diencephalon, and this sort of outcropping from that extends as this sort of C-shaped cup called the optic cup, and eventually this becomes the stalk becomes the optic nerve. So the optic stalk becomes the optic nerve, and then the optic cup eventually turns into the retina, which is the surface at the back of your eye that contains light sensing cells. And we'll be talking about it a lot more later. But what's important here, what's interesting here, is that the retina technically is part of your brain. So since it grows straight out of the neural tube, we consider the retina actually part of your brain. In fact, the retina is actually the only part of your brain, strictly speaking, that you can see from the outside without cutting a hole in your head. And then the other thing that grows out of the ventral surface of the forebrain is these little bulbs down here, called the olfactory bulbs. Now these are still inside the skull, but there are little nerves that come out of those that go into your nasal epithelium inside your nose, your nasal cavity, that you use to smell. So we'll talk about those more later as well. And then the next structure, if we go down towards the tail, is called the midbrain. The midbrain is probably the smallest of the three major divisions of the brain, at least in humans. And so the two main structures that form from that are the sort of dorsal half and ventral half. So of course here we're looking at the rostrocaudal axis, but when we look at it in cross section like this, you can see the tube that goes down the middle, which again is contiguous with the third ventricle, but we call it here the cerebral aqueduct. I guess just it's not big enough to warrant the name ventricle, but it really is just a small part of the ventricular system. And then the upper or dorsal part of the neural tube at this point we call the tectum. The word tectum literally means roof. And then tegmentum along the bottom, or the ventral surface, and the word tegmentum actually just means floor. So these two regions eventually grow into somewhat larger, more complicated structures. The tectum, which remember that's the dorsal surface of the midbrain, it eventually grows, and these four bumps start to grow on the top. The two bumps that are more anterior or above the other two, we call the superior colliculi. Superior, remember, just means above, and then colliculi is, or a colliculus is just the Latin name for hill or little hill, and that's all it means. They're just these little bumps that grow on the dorsal surface of the midbrain. So there's two that are more higher up, called the superior colliculi, and then the two lower ones we call the inferior colliculi. And here's what they look like in the adult brain. Now here what they've done is they've taken an adult brain and removed the cerebral cortex. So the whole cerebral cortex has been taken off, so you're looking at the dorsal surface of the brain without the cortex on top of it. You can see the thalamus, which remember that's part of the diencephalon. And then right in the middle you have the two regions of the dorsal midbrain. So you have the superior colliculus, colliculi, so there's one side, a left and a right, and then the inferior colliculi just below those. And those are going to come up later when we talk about the visual system and the auditory system respectively. But those are the two main structures of the dorsal midbrain. And then the ventral half of the midbrain we call the tegmentum. And there are some important structures in there too. Mostly these nuclei, including the substantia nigra and the red nucleus, which are important for movement. Also this periaqueductal gray, which is important for a number of different functions, including its involvement. It has some involvement in the autonomic nervous system. Also important for your sense of pain, actually, and we'll talk about that later on. The other major thing in the ventral midbrain are these big white structures here, white matter structures. These are basically, they're called the cerebral peduncles, but they carry their axons in there that are going from the cerebral cortex all the way down to the spinal cord. And so those are the only real important structures in the midbrain. And again, we'll come back to some of these as well later on. The hindbrain, meanwhile, this is it's called this because it's the anterior-most part of the neural tube that becomes the brain. So first of all, the empty part inside the hindbrain, this part of the neural tube, we call it the fourth ventricle. So fourth ventricle sits right here in the middle of the hindbrain. And as the rest of the hindbrain grows, the major thing that happens is on the dorsal side of the hindbrain, these two large structures called rhombic lips start to grow. And as they get bigger and bigger, they eventually meet in the middle and they form the cerebellum. So the cerebellum is this big, relatively big structure that forms on the dorsal surface of the hindbrain with the fourth ventricle underneath it. And then at the very bottom, the ventralmost part of the hindbrain forms this structure called the pons. And so those are two of the parts of the hindbrain. You can see here in the adult brain what it looks like. Again, the cerebellum is the only other part of the brain other than the cerebral cortex, which is easy to spot from the outer surface of the brain. And then the pons is usually easy to spot because in cross section, in a sagittal section like this, the pons is this thing right here. So this is the pons that kind of sticks out underneath the cerebellum. And again, in cross section, you can see it's just this big cluster of what we call pontine nuclei. And the pontine nuclei have functions in a lot of different areas that we'll talk about, a lot of different systems. So there are some relay nuclei for the sensory systems. They're also important in movement and a bunch of stuff that we'll learn about later. And then the other thing that forms as part of the hindbrain is the medulla. So the medulla is just what forms posterior to the pons, and it's the posterior most part of the brain before you get into the spinal cord. And the way it develops, the fourth ventricle actually ends up kind of sitting on top of the medulla with a very thin layer of meninges above it. And in fact, between the medulla and the pons, there are also little ports where the ventricular system lets CSF out into the subarachnoid space. But the medulla in the adult brain is, again, just sort of this simple looking structure that consists of a bunch of different types of nuclei. And a lot of these are, again, important relay nuclei, like the inferior olive is an important relay nucleus for the auditory system. This structure right here, nucleus of the solitary tract, is also called the gustatory nucleus because it takes input from the tongue. Vestibular nucleus is an input nucleus for the vestibular system, which we'll talk about. So we'll get to all these later. And then there's also a bunch of important white matter pathways, like the medial lemniscus is an important pathway for the somatosensory system, as is this structure here, the dorsal column nucleus. And so when we get to the various sensory systems, we'll come back and talk about some of these structures. Now right here, this cross section here, this is probably right at the boundary where the medulla starts to turn into the spinal cord. So instead of the fourth ventricle kind of sitting on top, that's how you can tell you're in the medulla, is you have the fourth ventricle kind of sitting on top of everything. And then as you get into the spinal cord, the fourth ventricle becomes a tiny little tube that moves down toward the center of the structure, and so we call it the spinal canal. And so that's where you kind of know that you're crossing over from what is formerly the brain to what is formerly the spinal cord. And then, of course, the spinal cord itself is the posterior most part of the neural tube, and it's probably the simplest in terms of structure because it essentially just stays as a long tube. So even though the brain has all these swellings and things that become convoluted as they grow and develop, the spinal cord basically retains its shape overall from the embryonic state into the adult state. So when we talk about differentiation, that's what we mean, is when an embryonic structure sort of transforms into a more complex adult structure. So the spinal cord anatomy, again, is pretty simple. You have the spinal canal, which is just the the interior of the neural tube that extends down through the spinal cord. The gray matter of the spinal cord differentiates into 3 zones. You have the dorsal horns, which are on the dorsal side, and that's where sensory information comes in. So we'll talk about those in the context of the somatosensory systems later. The intermediate zone, which mostly contains interneurons. So those are going to be neurons that are important for communicating within the spinal cord. And then the ventral horn, which contains the motor neurons. So those are the neurons which are going to send signals or can have axons that connect to muscles. And so that's pretty much the basics of the spinal cord anatomy. And again, here's the spinal cord in the adult. And again, you can see the different regions and gray matter, and you can see the nerves coming in on both the dorsal and ventral side. The only other major structure are the white matter columns, and those are just bundles of axons that are either sending information to the brain or bringing information from the brain down. And we'll talk about those more later as well. This is just kind of an overview of the different parts of the neural tube. Now you're looking at it in cross-section. So again, the telencephalon is the anteriormost part that kind of folds back over the rest of the brain. And then the diencephalon, which eventually grows into the thalamus and hypothalamus. And then the midbrain, the dorsal side we call the tectum, the ventral side we call the tegmentum. And then the rhombencephalon, the dorsal side becomes the cerebellum, ventral side becomes the pons, and then posterior to that you have the medulla before finally getting to the spinal cord. Now, and then this is what those structures all look like in the adult brain. So again, the telencephalon is just all this stuff. This is all telencephalon here, all this, everything that you can see pretty much from the outside. Now here we're looking at a cross section, we're looking at a sagittal section, but this is all diencephalon, or telencephalon, I'm sorry. Diencephalon, meanwhile, consists of the thalamus and hypothalamus. So again, in cross section, we see here's the hypothalamus and thalamus. They have the thalamus in purple and the hypothalamus kind of in this teal color here. You can't really see the ventricles as much, but actually this whole space here is the third ventricle. So the thalamus is sort of the wall of the third ventricle. And then you have the midbrain. So the midbrain again has 2 sides. We have the tectum up here, and then the tegmentum down here, and then the cerebral aqueduct running down the middle. So here's the cerebral aqueduct between those 2. And then the hindbrain, again, here's the pons. The pons is usually pretty easy to spot. It's this big swollen part of the hindbrain. And then the cerebellum on top of that. And then this region here is the 4th ventricle. So the 4th ventricle kind of is sandwiched by the cerebellum and the pons. And then you have the medulla, and that's the end of the brain. And then everything after that is spinal cord. So, and then this is just kind of a schematic to show how everything connects to everything else. So again, the neural tube develops into these 4 main regions: the forebrain, midbrain, and hindbrain. Forebrain develops into the telencephalon and diencephalon. Telencephalon develops into the cerebral cortex and the basal telencephalon or basal forebrain. And the inside of the telencephalon we call the lateral ventricles. The diencephalon grows into the thalamus and the hypothalamus, and then the 3rd ventricle is inside of that. The midbrain grows into the tectum and the tegmentum, and then the cerebral aqueduct is the little tube within it. Hindbrain grows into the cerebellum and the pons, and then the medulla, and then the 4th ventricle sits inside underneath the cerebellum, and then the last part becomes the spinal cord. So if you're wondering how much of this you're going to need to know, basically these regions here, so the 4 main divisions of the neural tube, and then what they eventually grow into. We're not getting into too much detail right now because we're going to come back to a lot of these. We're going to come back and talk about the cerebral cortex a lot, especially as we get into the sensory systems. The thalamus also, and the tectum, will come up a lot when we talk about the sensory systems. The basal taillencephalon and the tegmentum, and then the cerebellum will also come up when we talk about the motor systems, and then the hypothalamus we'll talk about quite a bit when we get to the autonomic nervous system. So again, the important thing is to remember each of these major structures and how they develop, or what they develop from in the neural tube. Now, before I wrap up here, I want to say a few things about how the brain is, the human brain is different from that of other animals. And so, first of all, before we talk about the human brain in particular, it's important to say some things about how the mammalian brain is different from that of other vertebrates. So, in fact, the brain itself, the way the vertebrate brain is organized, is really unique. So, if you really, other animals besides vertebrates have brains, but the vertebrate brain is kind of special. Generally, it's the largest and kind of most centralized, and it always has the same three parts. So these three divisions that we talked about, so the forebrain, midbrain, hindbrain, exist in pretty much all vertebrates. All vertebrates have those same three regions. The only thing that makes them different is how each of those regions grows and which ones become larger in the adult brain. So if you compare, for example, looking at fish brains versus other brains, you can see that the forebrain, so the forebrain is kind of colored in red and orange up here, is relatively small. So the forebrain is the smallest part of the fish brain. As you, in fact, if you look at the evolution from towards mammals, basically it's characterized by a extreme enlargement of the forebrain. So even in amphibians, so probably in amphibians about roughly a third of the brain is made up of forebrain. In the reptile brain, it's roughly half. Birds, the forebrain still is pretty much only about half of the total brain volume, whereas in the mammal brain, the forebrain makes up well over half of the total brain volume. Meanwhile, though, in other brains, and also it's important to point out these are not drawn to scale. So the fish brain here, it looks like it's about the same size as the mammal brain, but in reality, the fish brain is a teeny tiny, just a few, it would probably fit inside just one little olfactory bulb in the mammal brain. But it's important to show them at the same size here to show how enlarged or how the different structures are enlarged relative to each other. So in the bony fish brain, for example, the tectum actually is the biggest part of the brain, and that actually holds in other species as well. So even in amphibians and reptiles, still the tectum makes up about a third of brain volume, and even in birds, you still have a highly enlarged tectum. And the tectum, in fact, in these animals, is sometimes called the optic lobe. So the tectum's in green here, and so the optic lobe, the reason it's called that is because it's very important for the development or for the function of the visual system. Whereas in mammals, a lot of that function, instead of happening in the tectum, happens in the cerebral cortex. So that's probably one of the reasons that the mammalian tectum is proportionally so much smaller. Of course, the other thing that mammal is enlarged in the mammal brain is the cerebellum. So the cerebellum is a pretty big structure in the mammal brain compared to that of other vertebrates. In fish, the cerebellum is basically just a little bump on the dorsal surface of the hindbrain. And then, so the cerebellum, as we'll learn later, is really important for complex movement. So that may be the reason why mammals are so much better at certain types of movements than other vertebrates. The thing that the mammal brain doesn't have, another thing that the mammal brain doesn't have, however, are these big olfactory bulbs. So the olfactory bulb is, in fact, one of the biggest parts of the brain. If you look at a fish or, say, an amphibian, and again, even in reptiles, birds, the olfactory bulbs are these very long structures. Now, in some mammals, the olfactory bulbs are relatively large, but overall, the olfactory system is probably less important for mammalian as a mammalian sensory system than it is in other vertebrates. So then if you look specifically at the human brain compared to other mammals, one thing to point out, and we'll see this more in a second, everything in the human brain you can find in every other mammal brain. And in fact, when we look at pictures of brains or when we study brains, oftentimes we're studying those brains of other animals. In fact, a lot of what we know about the human brain and brains in general comes from studying rats or monkeys or other species, because of course there's some experiments you just can't do with humans. So we actually use these other mammal brains sometimes as models for human brains. Again, the only thing that makes the human brain different from other mammals is one size. So here you can see a bunch of different brains that are all scaled up to be about the same size. But you can see over here on the left the scale of how they actually appear next to each other. So way down here you have a teeny tiny rat brain, and then the human brain is at the bottom of this list. And of course humans have bigger brains than most other animals, but we don't have the biggest brains. So larger animals like dolphins, whales, and elephants have brains that are quite a bit larger than ours actually. But what is important is that our brains are disproportionately larger. So that means that the proportion of our that the brain takes up in our body and the size compared to what you would expect for an animal our size is much larger too. So humans don't have the biggest brains, but we do have the biggest brains for our bodies, or one of the biggest anyway. And that probably is one of the reasons why human cognitive function is more advanced than other animals, but there are other reasons why that might be as well. But like I said, size aside, what really makes the brain of a human different from that of other mammals is not that we have parts of our brain that other animals don't have. It's just that the parts we do have proportional to each other are much bigger, especially the cerebral cortex. So if you look at, say, a rat brain versus a human brain. So obviously the rat brain in absolute terms is much, much smaller than a human brain. But if you scale up a rat brain next to a human brain, it really highlights how different regions have grown disproportionately. So the first thing you notice if you just look at the surface of the rat brain versus the human brain is the cerebral cortex it obviously has those bumps and grooves on it. So the surface of the rat brain is basically, of the cerebral hemispheres are basically smooth, whereas for a human there are all these sulci and gyri. And those are there probably to increase the amount of surface area of the brain. And the reason for that is the cerebral cortex, that we'll learn later, or we've already learned, that it has this layered structure, and that's kind of key to how it functions. And so having the brain kind of folded up allows there to be more cells basically in the same volume of skull, and as it would be without them. And that's probably what they're there for. So in fact, if your cerebral cortex was expanded to be smooth like a rat brain, you would have to blow it up like a balloon. So it would be, probably like the size of a beach ball in order to fit all that brain tissue into your skull. So the purpose of those folds is to just let there be more brain mass inside your skull without making your skull basically too large to support. But if you look inside the brain, you can see that the parts of a rat brain and our human brain are the same. So the rat brain has a forebrain and a midbrain and a hindbrain, just like the human does. And in fact, everything except the cerebral cortex is proportionally about the same. So if you look at if you just ignore the blue part of each of these diagrams, just look at the midbrain, hindbrain, and the cerebellum, the sizes are relatively the same compared to each other. So those structures, for the most part, are important for really basic functions. So when we talk about the brainstem and the spinal cord and the midbrain and even really the thalamus and hypothalamus, we'll learn that they're really important, but they really serve pretty pretty basic roles in relaying information to and from the cerebral cortex. And the cerebral cortex is where a lot more complex cognitive function happens. And so things like memory and language and attention, all of that happens in the cerebral cortex. And in fact, the only thing that really is disproportionately small in the human brain that's obvious from the outside is the olfactory bulbs. So again, the olfactory bulbs are these big structures on the anterior end of the rat brain. And in fact, most other mammals have a relatively large olfactory bulb compared to humans. So for a human, the olfactory bulb is just a little teeny tiny bump on the ventral surface of the brain. And that reflects the fact that humans, and in fact, actually all primates, rely a lot less on our sense of smell than other mammals do. So as we'll learn, humans and primates are much more visual in terms of their sensory systems. And so that's why probably the olfactory bulb is so much smaller in humans than in other mammals. All right, so next time we'll talk about some of the techniques that we use to image the brain.